Formal part. Welcome everybody to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night by Zoom, 50 times a year. Tonight, it's my pleasure to welcome back to Wednesday Night at the Lab, Dave Lovelace of the Geosciences Department and the UW Geology Museum. Dave, here come the 5 questions.
All right.
Where were you born?
Well, I was born in Gainesville, Florida, on the University of Florida campus.
Very good. Were
Gator
your parents
by blood.
Well, that's reptilian blood.
Exactly. I thought it apropos.
Excellent. And were your parents working at the university, or do they have other connections with Gainesville?
Other connections. My mom was a neonatal nurse at the university hospital, and my dad taught at math at a local high school.
So what's it like when a neonatal nurse has a neonate?
What's it like?
Yeah, it's just kind of, never mind.
I assume, well, like everybody else's.
And where'd you go to high school?
I went, I really grew up in Casper, Wyoming. I was born in Florida, but at age 3, I moved to Wyoming, and I graduated from high school there after a brief sojourn to Colorado for a couple of years.
Very nice. And then where'd you go for your undergrad, and what did you study?
So I had this whole trajectory for my college career, and I wanted anything with UW in it. So I started at University of Wyoming, and I got an undergrad degree in geology, biology, and zoology, and then ended up staying for a master's where I studied biogeochemistry, looking at basically mineral and bacterial contact and some crazy stuff with proteins. So that was fun.
And then where'd you go for your advanced degree, or for your, for your PhD?
I decided I was done with that UW, and I transferred to this UW. And so I started my doctorate here in the geoscience department in 2008. And then in 2012, I graduated with my doctorate. And from there, moved on to working at the Geology Museum.
And what's your role there?
So I'm the research scientist in the museum.
So that means you find things and bring them back? And
yeah, so I spent a lot of time mentoring undergrad students and a few grad students, and we basically go to the field and collect material. Excuse me for just one moment. Sorry, my daughter was looking for her device, but it is not in here. So yeah, so I spend a lot of time with students working in the lab. We clean up fossils that we find, and then each of the students usually takes on an independent research project where they try to solve some problem with one of the fossils that we have, or working with the sediment that we collect.
And remind me, when was that area renovated? It seems like only yesterday to me, but
yeah, 2014 was when we started renovation. So we took what was the back room in the lab that had been kind of co-opted as a preparatory space for a number of years, and a lot of great work was done there, but it was literally done on tables that were scrounged out of dumpsters and anything you could find kind of on the cheap. And we were able to renovate that lab and really turn it around into kind of a professional lab with all the appropriate safety equipment and new equipment, which was fantastic, and really grateful for the university for that.
So when people come to the Geology Museum, and hopefully that'll be soon, maybe this fall, there's the big picture window. Is that correct? Is my memory correct?
Yeah. So there's a window that you can look through and see inside the lab and see all the activity going on. Absolutely. We are living goldfish inside there.
Way to go. How coy. Okay, so tonight you're going to talk about your work on the Triassic period, and really appreciate that. Why don't you go ahead and take it away anytime you'd like.
All righty. I am going to go ahead and share this screen then. Make sure I get the right one.
Yeah.
Okay. And all right. So just verifying this should be the title slide. Everybody can see that?
Looks good.
All right. Great. So welcome to Wednesday Night at the Lab. I am grateful to be here again. This is, it's always a pleasure. There's so many great folk that come through here and give presentations. So I am honored to be amongst them. So I'm going to talk a little bit about, or a lot about, the research projects that we've been really striving to work on and solve these problems, these issues out in the Triassic of Wyoming. And this starts as kind of an initial study for a unit of rocks, and I'll kind of explain where they fit in time and space. And we're trying to figure out what's going on with them. So a little bit of history. The Chugwater is the big unit of rocks that I work on out west. Now, when I was at the University of Wyoming, I had a number of professors in the geology department that whenever you got them really mad, they would force you to go work in the Triassic rocks of Wyoming because there's no trees, it's hot, there's no fossils, there's no oil. So nobody really studied them, and they would force us to go out there and try to find something exciting and bring that back. And of course, it would take weeks and weeks and weeks, and nobody would ever return. So that was the idea of their idea of punishment. And I thought, well, why? Why are there no fossils out here? So my initial interest or curiosity, what drove me into the Triassic of Wyoming, was trying to figure out why there are no fossils in this unit of rock, whereas the units that are comparable in age down in the Four Corners area are chock full of fossils. And they've been well known for the last 100 years. They're really well studied, beautiful outcrops. So that kind of drove my interest, trying to understand what it is. And that was really the focus of my dissertation work. And that was generally focused in the early Triassic. And then I ended up moving up section into the late Triassic. Now, to give you an idea, so here's our crew. We explore basically anywhere there's an outcrop of late Triassic rock exposed in Wyoming, we try to explore it. And although we're going for a rock ballad, I was trying to, make our own Abbey Road. Didn't quite work out for us. Our white album ended up being a brick red album. But besides working out in these really beautiful, wonderful, photogenic areas, it can be a little bit hot and it's dry, as we alluded to earlier. But I love working out west. I love working in those rocks. So the Triassic that we're studying, the time period that we're studying, is about 230 million years ago. And the Earth looked a lot different back then. And so if we look at the continents and how those continents were arranged in the Triassic, especially in the late Triassic, was during a time when all of the continents had merged together. This is what we called a peak accretion. So this is the time when all of the continents had finally smushed together into the supercontinent Pangaea. And the area that we work is right in here on kind of the central, near the equator. We're at about 10 to 15 degrees paleolatitude. And there's Wyoming. Okay, so that's the area that we're focusing on and where it sits within the continent. And as you can see, there's a couple things I want to point out. And the first one is the two little green lines that you see down here are actually remnants of the Ancestral Rockies. And so the Rocky Mountains lifted up about 300 million years ago to start with, and then they began to erode down and lots of rocks deposited around them. And then it wasn't until the very end of the dinosaurs at about 60 million years ago, 65, that the Laramide orogeny happens. And that was the big mountain building event that made the Rocky Mountains that we know and love today. So this is basically remnants of these old mountain cores sticking up, and that will play into our story later on down the line. I just want to point them out here. So here's the rock that is exposed at the surface that is of Triassic age in red here for all of North America. Mostly what I focus on is the rocks in the Western US, and then specifically the rocks in Wyoming. So if we look at our timeline over here on the right, we'll see the Mesozoic. This is the time of the dinosaurs. And the Triassic is the beginning of the dinosaurs. This, we call it the dawn of the dinosaurs. And really, it's a 50 million year chunk of time. And it's not until the last 30 million years of it that dinosaurs actually came into play. There were dinosaur-like animals, the dinosauromorphs. They're ancestors of dinosaurs, and a lot of other very cool critters. And most of those are actually the ancestors of all of the major clades. So the Triassic is a very, very important time period for the evolution of life on Earth, in part because at the end of the Permian, the period right before the Triassic, that's where we had the biggest mass extinction Earth has ever seen. 90+% of life went extinct. So the Triassic, the beginning of the Triassic, is actually a period where we're seeing a lot of recovery from this massive extinction event, and a lot of groups begin to diversify very rapidly. And so after that extinction event, we see things like the very first turtles, which we have the oldest turtle tracks in the world in Wyoming. And we have early mammal tracks at that same time period, and we have the first early mammals later in the Triassic. So mammals, where was I going? A lot of the modern amphibian groups originate there, and birds via dinosaurs originated in the Triassic. So most of the modern fauna that we see today, if you go out and you see an animal, most likely their ancestors, the actual origin of that group of organisms, originated in the Triassic. So it's a pretty pivotal time period for life on Earth. So here's the Triassic that we're looking at, that 50 million year chunk of time. And for most of this talk, we're going to be focusing on the last 30 million years or so. So here are the rocks in the continental United States, so the lower 48. And the rocks here in red, those are the chugwater group. So those are the rocks mostly exposed in Wyoming of Triassic age, and that's what we end up studying a lot. The green here is the Chinle Formation, and then the purple down here is the Dockham group of West Texas and eastern New Mexico. Now, later in the talk, we're going to focus on these three major groups of rock in the western US and see how they relate. Before this work, there really hasn't been much work to focus on correlating or trying to figure out that relationship. In fact, I always joked that the chugwater was the red-bedded stepchild of geology because it was often ignored. It was often just left on the shelf. It was the place people went to went to work to get punished. So it's often neglected, and I think I really wanted to understand why, and we're going to piece that together. And so these are the three localities in the western US, or in the chugwater, and then there's one little tiny place over here in Nova Scotia that I want to talk about, where there's some really fantastic specimens that came out of some exposures right along the coastline. And the beauty of those specimens in Nova Scotia is they actually have really nice dates that they're able to assign to those units of rock because of where they're located in Pangea, and there's some volcanic activity that allowed for really high resolution dating. So I'm going to start with one of the issues that I discovered during my dissertation work is that effectively, if you look at all the rocks in the western US and you kind of stack them up or look at them statistically, there is a huge gap in the rock record, and it's in the middle Triassic, and it extends all the way into the late Triassic to right here. And the problem is that in this time period, right where my cursor is at the bottom of the C, the Carnian, that's where dinosaurs evolved. So most of the early dinosaurs are known from South America. They aren't known from North America in that time period because there are no rocks that preserve them. At least that's what has been suspected for much of our history, geological history in the Western US. So with that, we'll talk about some of the rock in Wyoming that we studied, and I'm going to show one locality, and this is near Alcova Reservoir, and we'll just zoom through some of this. This is the rock that we see in the Alcova Reservoir exposures. There's a chunk of rock at the very top of it. It's a massive conglomerate, and there's some pretty important stuff comes from there, and even down lower. So In the low section, this is early Triassic, we have lots and lots of tracks, but no body fossils. And there are some extenuating circumstances, probably due to hyperaridity and super rapid deposition, that the bones get effectively diluted out, whereas tracks and impressions are much, much easier to preserve. But as you go up section, oops, go the right way. As you go up section, you start seeing more tracks, and eventually you start finding tracks and then bone. It's not until the uppermost part of this section that we start seeing bone, and this is a tooth of an archosaur, probably a phytosaur, which is a croc-like animal, sister to or related to dinosaurs, but not on the line to dinosaurs. And then up above it, we had this huge collection of these jaws. These are pieces of maxilla, the upper face bone, and some of the lower jaw of an animal called a Rankosaur. Now, when we first found these, we were super excited because, hey, there's bone. That was the first thing that was exciting. The second one was we had so many of these isolated little specimens. Now, these are found as class. So each one of these bones, look at the scale there, it's only about a centimeter long. So they're really small. But they're found as eroded pieces of rock in a massive conglomerate. So here's the rock that we're looking at, and you can imagine each one of these little pebbles is about the same size as this jaw fragment. And so we would find these little pieces of bone randomly throughout this conglomerate. And so very exciting, lots of jaws. And here's the Alcova Reservoir where we were studying it. And the reason that we found this locality is often what happens in geology is there's a little bit of serendipity. A little bit of special circumstance that just happens, luck of the draw, as it were. So we were sitting at this campsite enjoying our evening libations at the end of the day, and we weren't even doing field work here. This was literally a stop on the way to the area we were going to be spending the rest of our field season. So enjoying the outcrops here, sitting on our campsite. Literally, we look at the ground and one student says, hey, here's a little fragment of bone. I was like, oh, that's cool. Oh, yeah, here's another one. And I picked it up, and by golly, it was a little vertebra, but it was bone white. I assumed at that point that it was just a modern rabbit vertebra or something along that line. But I looked at it and I could see that the rock was actually embedded inside where the spinal cord would go. So I realized that this was actually a fossil, or it was a bone that came out of rock. So I presumed it was a fossil. We looked around and we started finding more and more of those fragments of bone in the conglomerate that we were camping on. And so that's what started this whole journey into trying to really understand what's going on with this specific interval. And so we continued to look out around these conglomerates and around rocks in the same time period, and we started finding more and more bone. So the last time that bone had really been found was in the 1940s. And prior to that, it was the 1920s. Ironically, the first and most impressive specimens that were found by the UW Geology Museum, or at least one of the members, Maurice Mell. He was the person who mounted our mastodon in the museum. And then shortly after finding bone, he moved to University of Missouri, and that's where the specimens reside that he collected. So we ended up going back out to the field, finding some new bone and new localities, and that kind of kicked us off. So here we are with this Rankosaur, and these are the jaws that we are in the process of describing right now. And so we'll be publishing these hopefully sometime later this fall. We'll be submitting the manuscript and see how that goes through review. But the Rankosaurs themselves are very informative in terms of their kind of location in space time. And so this is the animal that we're looking at, Rankosaurs. They have this kind of beak-like front. They're a little reptile running around, chewing on probably a little bit of everything. And let's see. So initially, the bones that were found in the Paposia, there was one other Rankosaur specimen that was found and identified, and then we ended up finding 14 more. So we've added a pretty good chunk of information to the little Rankosaur population of this area. And importantly, early on, they even noted that this is equivalent to Rankosaurs that were found globally. And they all were known to be from Carnian rocks. Remember, that's that chunk of time that we're missing in pretty much the entirety of the North American continent. So if we look at where these animals fit in kind of the evolutionary scheme, this is our phylogeny, which basically what we're seeing is that all of the specimens that we have found fit somewhere down in this range on the evolutionary scheme of things. There is a big group of Rankosaurs globally. Some are known from South America, Argentina, Brazil, and India, and the UK. And so these animals are all very closely related together, very closely related, and ours sit just outside of it. Okay, and the green dots that you see here are the associations of hyperidapodons, these little Rankosaurs that all go extinct, notice, by the end of the Carnian. Those ones with the green dots are the ones known to be found with a certain species or certain group of Phytosaurs, these croc-like critters. And same tree. This is the specimen that it basically is just a finer resolution. We're looking at most of our specimens fall within this group that we're naming, and it is sister to this one, Ornithorhynchus, and that is the specimen from Nova Scotia, which we'll see a little bit later in the talk. So there we go. We found the Rankosaurs, and at this point, this was early on in my graduate career. We came back. Our family started to grow. We headed back out west every summer, and the kids kind of got to grow up out there and spend their summers camping, in horse tanks. It was it's always an exciting time. So what kicked our first field season with students, where we had a number of students coming out with us, I looked at Google Earth and I was like, I know where I want to go look. There's a road map and it says Madison and Chugwater. And so I thought, sure, of course, where Madison meets Chugwater, that's the first place we have to go look. And here's the rock crew, our band, as it were, sitting out here on the rocks after we had found this Phytosaur. This is the first Phytosaur that has been found since the 1950s. So these two women down here were Mel and Kim. They found the first fragments of this Phytosaur, and they were tiny. And it turns out what they ended up finding was just the tip of this snout. And there was about 6 pieces, and we were able to put it together. And I'm like, oh my gosh, it's like the tip of a snout. And you could see the teeth coming off. It was really cool. Well, they ended up spending the next 20, 30 minutes kind of working their way up about 10, 15 meters of the mountainside. And they found what was probably the size of a kiwi was the cross section of the snout sticking out of the rock. That was all they saw. And I was very impressed that they found that. We ended up uncovering the rest of the skull and excavating it in the course of 2 or 3 days. And that is what kicked off our super excitement for looking at the Triassic. So this is where we were actually intending to head that first year that we got sidetracked with all the Rankosaur material. And we're starting to realize there's actually bone out here. There's actually more to this story. So each of these little puzzle pieces is starting to flesh out a little bit more of our understanding of what's going on out in the Chugwater. So with each of these specimens, we take them back to the lab. We clean off all of the rock out of them. I'll go back real quick. So realize this Phytosaur, whenever it came back, was completely encased in rock. And it took us over a year for one of our volunteers, Brandon Price, and myself to completely prepare this specimen out of the rock. It takes a lot of time to extricate this bone just to study it. So after a year's worth of preparation, we took the skulls into the WIMR on campus, the Wisconsin Institute of Medical Research, or imaging. So we imaged the fossils using CT scans. And map out things like sinus cavities and where the brain was, and understand how big the brain would have been and what the nerves would have looked like. So it's a really informative tool for basically being superheroes and looking through rock. And it's a really especially useful tool for understanding kind of the biology and the physiology of these animals. So turns out, remember that 1940 being the last Phytosaurs found? Well, this was found in 1948, and this is a specimen that was sitting on a shelf in the Field Museum. And somebody came by and described the specimen just like this. They didn't take the time to prepare it. And so we borrowed the specimen from the Field Museum, and then they sent along this big chunk of rock as well. And they're like, we have no idea what's in here, but it has the same field number, so it probably came from the same site. So Brandon and I and a couple other students spent some time, prepared this fossil, and lo and behold, it's another of these Phytosaurs. So then we, and it's a beautiful specimen. So what was in that chunk of rock is actually this specimen. It was the snout of that critter. And so we're in the process of redescribing these two specimens, or describing this one and redescribing the Field Museum specimen. Notice this kink in the nose right here. This middle section is actually rock. That's a fault. So where the rocks slid past each other. But we decided to leave it in the specimen because it's kind of a cool little piece of the story of the preservation of this critter. So here's our Phytosaurs, these croc-like animals. Again, we do the phylogeny. We try to put these into their evolutionary place, kind of the family tree of these critters. And what I want to point out is, unlike our Rankosaurs, who are related to the most derived, they're all the way up at the top of the tree of Rankosaurs before they go extinct. Our Phytosaurs that we have are actually the most primitive, the most basal group of Phytosaurs known from the whole entire clade. And so here we can see the green dots, the Hyperidapodon. That's where all of the Hyperidapodon specimens, little Rankosaurs, where they co-occur with these Parasuchids, these basal Phytosaurs. And again, it's that Hyperidapodon, the fact that these little Rankosaurs have some sort of temporal constraint geologically that's really helped us. So if we look at where those Rankosaurs are found globally, along with their Parasuchid counterparts, we see that in the Esquilasto in Argentina and the Parana Basin in Brazil, we see a number of specimens, Tyambaida and Hyperidapodon, from these units, and they're all older than 200, bound somewhere around 230 near the base. So they're within this 5 or 6 million, constrained by both the radiometric dates in Argentina and Brazil.
Dave,
Other
if I
dates
could interrupt a little bit, your
Yeah.
audio is tending to go really low and then come back. If there's any wires you could check for your audio, I'd appreciate it. Thanks.
Absolutely. What I'm going to do, I think, is see if I can do. I'm going to try this, Tom, and let me know if this works better. All right. Can you hear me now?
Yeah, but it sounds like you're in a barrel.
Yeah. Okay.
So it was weird because it was going.
Go back with the headset and see if this will work.
Yep. We'll stick with that. Thank you.
Yep. So the Hyperidapodon is basically bound by these radiometric dates that help us constrain the age. And so we're using that kind of as the linchpin to help move our units of rock in time and space. So the Phytosaurs that were found are also able to be constrained because of the similar groups that are found in Germany. So you have Hyperidapodon and these Parasuchids that are known to be from Carnian rocks because they're bound by marine strata that have they're related to ashes out in the ocean. And so we're able to constrain Parasuchids based on those specimens and then the Hyperidapodons. So all of this is pointing to Carnian aged rocks, which is, again, pretty darn cool because there aren't really any Carnian rocks known from North America. This is another site. It's a tiny little outcrop of Triassic that is surrounded by a sea of Eocene rock. So basically, when the basin filled in, it filled in with all this Eocene aged, about 40 to 50 million year old rock. So this is after or during the process of the mountains building up, the Rocky Mountains. And as those rocks, those Eocene rocks, began to erode, it exposed this tiny little window of Triassic. And so we went out there because I found it on Google Earth, and hey, why not? Let's go explore it. And turns out this little site, this small little outcrop, this is all we got. And there were a number of small fragmentary bones that we found on the surface. And we spent a number of days screening and really trying to find everything we could find on that surface of this pretty exquisite specimen. And I will get into this at the end. This is kind of the culmination of everything, was what we ended. So we collected enough of this material, brought it back, began to prepare it and catalog it and identify it. At the same time, we were working in rocks just outside of DuBois, Wyoming, and we are literally parking our cars at the end of 3rd Street in DuBois, and we walked out on some BLM land, and there's 2 quarries here that are pretty expansive, and this is where for me, it was justification. All of this stuff has been great, but it's all been pretty fragmentary outside of the Phytosaur skull. It wasn't until Knobby Knob was discovered. We went out, excavated, and the students we spent 4 years now, 4 seasons working this quarry. And part of the reason is this is a mass death assemblage, but it's at the very base of the Paposia Formation. And here is one of the dozens of blocks that we were able to pull out. And as you can see, each of these, this is one skull right here. This is the nostril. This is an orbit. That's the other eyeball. This is a skull that's upside down. There's the eyes here. You can see little tiny teeth. Here's another skull upside down. And then there's another one over here. So this is a mass death assemblage, just meaning there's a bunch of individuals that have been collected and prepared from, slowly being prepared. Aaron Kufner has been, he was an undergrad at the UW and now is a PhD student, just got dissertator status, and he is focusing a lot of his research on these animals and kind of, again, placing them in their evolutionary context and understanding more about their biology. So one of the interesting parts of this quarry is the preservation is phenomenal. And this is not something that we get very often in paleontology as a whole, let alone in the late Triassic rocks of Wyoming. So as we zoom in on the inside of the skull, this is its palate on the right-hand side. And this is basically right back in the back of its throat. And if we zoom in here, see these little bone fragments. As we began to clean and uncover them, this is what we find. These are palatal teeth. They're little tiny teeth on the inside of the soft palate. So they're actually stuck inside the skin. And this is one of the ways that the organism is able to intake and swallow food, much like a frog would or a similar animal. And so the fact that these are preserved in situ, like in place in the skull, is amazing. It's one of the few one of the few sites that preserves this for this whole entire clade, which is really great. So again, all this information from these amphibians is starting to put us in another context, which is within this amphibian group. If we look at their eyes, there's a bone. This gets into the kind of nitty gritty of how we identify species and how they relate. This group of Metopasaurs, really interesting because it lacks, this bone right here is a lacrimal, and it does not contribute to the orbit. It's outside of the orbit. Now, as you get higher in section, there's another Metopasaur called Anaschisma, and that one actually has this bone. The lacrimal is part of the eye, part of the orbit. And so there's a separation in this group based on where you are. Evolutionarily, it seems that this one character is a big deal. We don't know why. And that's one of the things that Aaron is studying. Now, our animals happen to be in this size class down here, the smaller of the Metopasaurs. As you go up section in the Paposia near the top, they get to be much, much larger. And so we don't know, and it's one of the things, again, Aaron's looking at is this an age class? Are we seeing the bigger ones are older, or are we seeing some sort of difference in kind of the evolutionary history? The ones that we find up section do have this bone included in the orbit, whereas this one doesn't. So there's something going on, and that's one of the things we're actively looking at right now. But it's almost a population level study because we have so many of these specimens. All right, so just about 150 meters from Knobby Knob, we found another site. And this is a riverbed that whenever you look at the strata, basically this riverbed has a whole bunch of vertical burrows in it. And within those burrows, we're finding these tiny little amphibians. So they're similar in that they're closely related to the other Metopasaurs, the huge ones that we're finding. But these are like super tiny. They fit inside a Pringles can. And here's a cross section of a skeleton that we found. These are just sitting on the outcrop. They're all over. So we ended up collecting a number of the burrows, and we sent them down to the one of the nuclear reactors in Australia. And colleagues there have scanned these specimens using high-powered electron beams, looking at synchrotron and neutron scans, basically really super powerful CT scans. And one of our other students, Calvin So, who was an undergrad in Madison and now is doing his PhD at George Washington University, is helping us identify and study these animals. So a lot of the students that went with me to the field are actually actively continuing on with the research and using this as kind of the foundation of their advanced research. Now, this specimen is another one that we sent down to have synchrotron scanned in Australia. And it is the whole entire specimen is the same length as one of the tiny skulls. So this is a super tiny critter. And it has the whole skeleton in there, but no skull. However, the whole skeleton is there, and that's been super informative and helping us place these critters kind of into, again, their evolutionary context. And there happens to be another small little amphibian very closely related to this that's found at the very base of a unit in Texas. So we'll kind of start looking at rock and comparing these things and putting this story together and figure out where we are and what all we can say about that. Okay, so let's go back. So originally, when I first started in paleontology, the typical thought for the Chinle and Dockham and the Chugwater was that these lower units at the base, the Shinarump, which is a massive conglomerate, is the same as the Santa Rosa, is the same as the Paposia. So they were all considered to be lateral equivalents. And then a number of radiometric dates were determined for rocks throughout the Chinle group. And much of the formations of the Dockham followed the Chinle as we were able to constrain it within time. So we have a much better idea of what those rocks represent in terms of time, as well as those in the Dockham. The problem is that some of the rocks in the lowest Dockham they followed with the Chinle, but then there were some oddities and people were like, we don't know, and kind of left it at that. But then the Chugwater was left floating. Nobody really knew if that went up to or how that changed. So the Chugwater for much of the last couple decades has been, once again, sitting out in space with no idea of where exactly it fits in the time frame here. Enter in tiny little crystals. So these, anytime you get like a volcanic event and the ash goes out, they'll often have little zircons incorporated in them. And those zircons are a great clock for the timing of that volcanic event. So ideally, you have an ash that gets deposited, and that starts the clock in these crystals, and that gives us an idea of how old that rock is that contains that ash. Unfortunately, ashes are extremely rare. So that material gets transported into a river and then deposited in sandstones. Sometimes that can happen at the same time, so it's really still recording the same age as the crystal. Sometimes those things get picked up from rock that are millions or hundreds of millions of years older, or even billions of years older, and redeposited in much younger rock. So the geologist's job is to interpret all of these zircons and try to get an idea of where did they come from and do they represent what we call the youngest, or the way to get a maximum depositional age, so that we know that the rock is no older than whatever the zircons tell us. So these studies, we're looking at hundreds and hundreds of zircons, and what it does is it gives you a spectrum, right? A probability distribution of ages from all of those zircons. And usually whenever you hit a you get a couple that are really young, and you're hoping that that helps you to start constraining ages of rock. Now, the study wasn't even looking at ages of rock. All it was really looking at is the genetic relationship. So not genes as we think of organisms, but how related are these rocks to one another based on where the sediment came from, the source area. So if we look at these, this is the Shinneramp, right? That's that massive conglomerate. The very base of the Chinle and the base of the Dockum has similar groups called the Tres Lagunas of the Santa Rosa. Now, look very, very similar. And in fact, they all come from what has often been considered the main Chinle trunk of the Paleo River of the Chinle Formation. So it's this large river system that's moving across the landscape. It incises down into older Triassic rock, and it starts late Triassic deposition. Now, if we compare that Tres Laguna specimen, which is what is equivalent to the Shinneramp, and we look at another group of spectra, which are three outliers, three oddballs. One came from the sandstone, the Tecolito. One came from the Gartra, which is the basal conglomerate in the Paposia. And it turns out that those two seem to be extremely similar. And then in the Santa Rosa, there's another one from Texas, and then the Gartra from northern Utah. And with that, they started to speculate. So these authors speculated that the differences in the genetics of these major sandstones shows that there was an evolution of three rivers over a course of time. And if we look at the rocks themselves, this is our new hypothesis based on those zircons. It seems that it's pulling these units apart and starting to separate them. Now, exactly what this duration of time is, we don't know. So that's our question mark, and that's what we're trying to figure out. So that kind of sets up the geological scenario. So here we have these three discrete groups of rock, and they always start with a high energy conglomerate at the base, and then they go up into finer grain mudstones above it. And that is typical of basin evolution. You start with high energy as you incise into the basin, you deposit out your rivers, things start to fill up, and your energy decreases, and you get more and more fine grained sediments. Notice that we have three of those discrete packages where you have this coarse base, we have a fine sequence up above, and same thing up here. We have that same coarse base, and then up here, coarse base in mudstones up above. There's actually a fourth unit in the Triassic, but we're ignoring that for right now. This stuff is very well constrained temporally, and that's what we're trying to go back. So these are the depositions. These are the rocks that are preserved in the Dockum of this age, and the river probably flowed down into the Gartra and Paposia on this side. And then what happens is the river jumps course. Of course, you can't see my hands, but imagine this river is jumping to another location, and it incises down here, and it begins the deposition of the classic four corners Triassic rocks that are so beautiful, like petrified forest. And then the river jumps course yet again, and another series of sediment evolves out of that. So as the sediment continues on, building throughout time. We come back, we're looking at these chunks of rock that seem to be slightly different ages, and we start putting the biology, right, the biological organisms into those rocks and seeing what the biology or the paleontology is going to tell us about the relationships of these rocks, or if it helps support it. And so remember, these are the Phytosaurs. We're going to look at Leptosuchus here and Parasuchus. So I want you to remember those two critters specifically. We're going to go back to our phylogeny and notice here's all of our Parasuchids in the red here from the Paposia Formation. There's other Parasuchids from India and some from Germany. But the Leptosuchids are up here, higher up in section. They're actually found well above. There's none found with Hyperidapodon. These are the lowest Phytosaurs found in the Chinle. Okay, so if you go all the way to the base of the Chinle sediments, the very first Phytosaurs you find are Leptosuchids. So again, going back here, so here's Leptosuchus where we find it, and then Parasuchids come from this lower unit. Along with that, we tie in the whole range of Parasuchids is actually pretty extensive. It goes into the Norian, right in here, about 225 to 220. But the presence of our Rankosaurs pulls it down back into the Carnian, and it constrains it here. So we have the overlap between these critters that span into younger ages, and we have critters that span into much older. And it's where those two ages overlap that it's helping us to constrain the timing of the deposition of the Paposia. And notice right next to it is this non-lacrimal orbit. Again, that's that little Metopasaur that the bone is outside of the eye socket. And then the Metopasaurs that have it, that it is part of their eye socket, begin up above it. And so again, we can use those to constrain it because we have both the Metopasaur from Nova Scotia, the Rankosaur from Nova Scotia, that are constrained with other dates. And so we're able to tie all of this stuff together between our Phytosaur, our Metopasaurs, and our Rankosaurs, and say that we truly have Carnian aged rock. So here's the Metopasaur from Nova Scotia, from the Wolfville Formation. Again, the lacrimal sits outside of the eye, just like our little guy down here. So when we compare all of these animals from the Paposia, our whole ecosystem that we've collected since about 2012 was when we started. So really over the last 8 years, between collecting and preparing and studying these specimens, we're starting to get a pretty good idea that we have, of the animals around, and that they compare extremely well with this group down in Texas. And so that's our hypothesis that we're putting forward, that these two units of rock are equivalent. And because of the presence of both Parasuchus and the Rankosaurs and the species of Metopasaur, that we have some reason to assign all of these rocks to a unit of time. And so it fits within the Carnian. So that's our hypothesis of where these faunas fit and the ages we associate. So these are our rocks, the Paposia, and those are the non-Leptosuchid Parasuchids, right? So that's all the Parasuchids. And then the first Anaschisma, which is the first Metopasaur that has the little bone in the eye, sets up the second chunk of time, and then our first Leptosuchids come around. So it's within this chunk of time that are right at this break, these two, the red and the blue, that our rocks are situated. So with that, if you'll recall that one little outcrop where I said there was a couple cool little bones that were found, they were neat, we took them back and identified them. It was this bone right here and this one right here. That is the upper leg bone, the top of the femur, and this is the ankle bone. Those two bones are diagnostic to dinosaurs. So the collection of bones that we have here are actually representative of the oldest dinosaur in North America, at least, probably the Northern Hemisphere. And that it really helps fill in some gaps with our understanding of dinosaur evolution and dinosaur distribution around the globe. So if we look at where dinosaurs fit in, our dinosaur fits in the big scheme of things, Chugwater, again, has the oldest dinosaur down here. The next oldest one is actually from this chunk of rock down here in the Colorado City Formation of the Dockham group. However, it is found with those Metopasaurs with the bone, the lacrimal, inside the orbit. So it's in that blue zone. And most of the classic dinosaurs like Coelophysis, Chindosaurus, et cetera, are from these upper rocks in the classic petrified forest Triassic rocks of the Southwest. So with that, if we look at the relationship of the dinosaurs, I want to show you a couple interesting features. Is that these dinosaurs down here are the oldest known in the world right now. And there's a couple in Brazil that are pushing this down a little bit further. But within the dinosaurs, most of these come from Brazil and Argentina. Our taxon fits somewhere in this zone, and it might be very close to the top. So we cannot claim we don't have that fine of control, that higher resolution. But we're able to at least say it's amongst the oldest dinosaurs in the world, which is pretty spectacular. Even if it is a partial, relatively incomplete specimen, it still is evidence of the earliest dinosaurs on our continent. And so these are the position of the remainder of the dinosaurs that are found phylogenetically. But as you can see, our little guy sits right outside of the true theropods, the neotheropods, and within the early theropods, including stuff like Eodromius and Herrerasaurus. So it's closely related to neotheropods, but not quite there. So it's kind of at the base of it, which is really interesting in terms of understanding the evolution of theropods and how they're distributed around the world. So with that, I just want to say, all of the students that are working out in the field, my parents who go out to the field with me, honestly, it's an awesome privilege and pleasure to be able to go out with my family and my students and work on these rocks. It's a lot of fun. And the specimens that we found over the years, these Phytosaurs, the little amphibians, and the little Rankosaurs are all super important. Each one of those little pieces has added to our whole picture of understanding kind of a time and place for these rocks. We have sent off our own samples of sediment off to Idaho, where they specialize in looking at these groups of zircons. And we're hoping that sometime in the next few months, we might actually have our own radiometric age to help test this hypothesis, which would be great to have further support for a Carnian age. The dinosaur also is being written up currently and is getting ready to be published along with the Rankosaurs. So our assemblage is certainly older than the rocks that are typically seen in the Southwest, which is pushing our Triassic coverage into the Carnian for the first time. And lo and behold, the Paposia has dinosaurs, which is pretty spectacular. So we're very excited about all the future work that's going on. There's a number of new localities we'll be exploring. And of course, spending more time just walking outcrop, looking for new sites and seeing what else the Triassic of Wyoming has in store for us. So with that, thank you very much for your time. And I am happy to answer any questions you may have.
Thank you very much. What we tend to do, Dave, is to ask people if they want to put their questions in the chat, and Bea tends to monitor those and to read those, or people can chime in. And if people are willing to chime in, then we alternate spoken with a written or read one. Would anybody like to start with a spoken question for Dave? If not, Bea, why don't you read one of the first ones then, please?
Yeah, the first question in the chat is from Sandy, and who asked, any comments on the New York Times article, Archaeologists versus Computers, a study test who's best at sifting the past? And it was just posted yesterday.
Oh, wow.
So a real recent article. He has a link in there too.
So I'll answer the question, sort of. No is the short answer. I have not read the article. However, there's two things. First, archaeologists versus computers. I think one of the things that they're using is, and I don't know this article specifically, but I know that they're using some AI and pretty cool computer tools, basically, to look at a whole collection of rocks or pieces. And they see if the computer can look at those and match them up better than people can. And that's to archaeology specifically, and it may be applicable for sure if that's the case to paleontology, but slightly different field. However, there's another thing that has been recently used, which is Google Earth, where they use AI. Basically, they go in and say paleontologists have scoured a particular landscape, and the paleontologists come back and circle, hey, we know there's fossils here. We've surveyed this area over here, no fossils. This one, yes. And so all of those places that have fossils get marked. And then the computer comes in and says, okay, when I see sediment patterns or packages that look like this, I'm going to flag it. And so then they go out and they flag these potential areas to go look. And it's a pretty high hit rate. It's certainly not perfect, but it is better than random, going out. It actually has a predictive power, which is pretty interesting, and it has a lot of potential for paleontology. However, because they're testing this, their ground truthing those exercises by going to the field and looking, it still has the human error of they can go to the field and look at an area and not find something, or areas that the computer predicts that there aren't fossils, people can go out and find things. Like the Triassic Wyoming, where forever people said, oh, there's no fossils out there. It turns out there are fossils. You just have to look really hard and spend a lot of time looking. So I'll have to look at that article, but thank you for sharing that link, Sandy.
Sounds like a pretty powerful sieve. At least it gives you a better shot at where to.
Absolutely.
Would anyone like to speak a question? If not, Bea, go ahead with the next one.
This one is from Tom, but he asks, what kind of tools, hand or power, do you use to excavate the fossils from the rock?
Cool, good question. So we have, it's really funny. So I'm talking about all this AI and really fancy computer stuff. And really, the actual excavation hasn't changed dramatically since the 1800s. We use big hammers and chisels, all the way down to just little dental picks and toothbrushes. So most of our stuff, we remove the rock with picks until you get close to the bone, and then you have to determine where the bone rock contact is. Basically, you're finding the edges. The goal is to find the edges without removing too much rock. The more rock that stays on the specimen, the safer the specimen is. It's been in the rock for 230 million years. And as soon as we start taking it out, it's going to begin degrading, not just because of air or anything, but just because we're chipping rock off of it. And so we want to do that in a controlled a manner as possible. And so once we find the edges, then we go even farther out, and then we use big picks, hammers and stuff, and break up the rock until we can get a jacket around the rock encased bone. And that jacket holds all the rock together, and then we dig around it and basically flip it over on its side. So all of those techniques literally were done in the 1800s when they were excavating the very first dinosaurs out west.
And then when you get those back to your lab, how do you, what kind of tools do you use in the lab to reveal the actual fossil away from the bone?
Sure. So there's a couple, we have two main sets of tools that we use. One, we can still use hammers and chisels to get it close to the bone, but as you get closer and closer, your tools change. And we have these little pneumatic, they're like tiny little jackhammers. It sounds like a dentist office when you go in there. It's just this high-pitched buzzing sound. And it's this micro jackhammer that's helping chip away rock. And usually the rock bone interface it allows for a pretty clean separation. And so you're able to buzz right up to it, and then little chip of rock will fly off, and there's your fresh, clean bone surface. And then you just keep doing that. It's just, it takes a long time to remove the rock slowly enough that when you get to that contact, you don't accidentally dive into the bone itself.
So you got to be good friends with an ophthalmologist.
That's right. Or we use really big microscopes or glass, magnifying glasses.
Wow. Any other questions that people would like to speak? If not, I'll go with the idea that people want to have Bia read them. So go ahead, Bia, and we'll just read the rest of the chat questions.
Yeah, so first Darius put in a comment on there. She says, OMG, the Field Museum let you prep the specimen. How cool. So very excited about that one. And then the next question is from Jim, who asked, did you used to have stuff scanned at the Aladdin Ring in Stoughton before that shut down?
I didn't. I wish that the timing, that I had known about it. It was shut down about 2 or 3 years before my first opportunity to, or before the thought crossed my mind to use this technology. And I don't know that they had synchrotron, but I don't know if they ever used or worked with fossil specimens. There's only a few facilities around the world. France, there's one on the East Coast that does take specimens. There's one in I think Baltimore that's coming online now, but it's actually our Australian colleagues that are training and helping the American institutions come online with these synchrotron scanning for fossils specifically. So soon I'll be able to spend more time in the US scanning specimens, but for now I use the Australian group, in part because the Australian government helps fund this open research, which is fantastic. And we're super grateful for it.
Very interesting that Australia is doing the funding. But
yeah.
Next question is the texture of the Metopasaurs relative or reflective of their skin? Sorry.
No, that's cool. So yeah, that texture that you see on the Metopasaurs, it's on their skulls, and they have these huge plates of bone in their pectoral girdle, their chest. So our clavicles, our collarbones, they have very big, huge clavicles and a special bone called an interclavicle. And those are textured in the same way, and we don't know exactly why. Whether the skin followed that topography, the bottom line is we don't really know. Crocodiles have similar texture on the scutes that go down their back, but you don't necessarily see that texture externally, not the little divots. Those end up being filled with soft tissue. So we aren't really sure exactly what they are. It's a great question because that is an active field of research and we don't have a good answer for it.
All right. I will keep going with the chat unless anybody wants to step in with a spoken question. Okay, so the next question, which lab in Idaho will date your zircons?
So I'm working with Mark Schmidt at State, and their geoscience department has an image or a facility where they specialize on zircons, and Mark Schmidt is kind of a worldwide expert on doing detrital zircon work and has all the cool toys. So,
and your audio kicked out right as you were saying the name of the place.
Oh, I'm sorry, Boise State.
Oh, the turf. Good. And isn't your department pretty famous for dating zircons or having a zircon that really is 4.2?
But it also so the Sims group works on zircons, but it's a little bit different than what we're doing. There's this uber high tech. They do some really cool work, especially with inclusions in zircons and looking at oxygen and various aspects. So we got cool tools too. But it's not the focus isn't looking at kind of these provenance or larger scale studies, and so we end up tending to go with labs that specialize in that. Although researchers coming into the department are starting to build labs that might give us that capacity. So.
Dareth in the chat if Indiana has a zircon lab, or she thinks Indiana has a zircon lab.
You know, it's there are a number of labs that work around the country that work with zircons, including UW-Madison. I'm not familiar with that one in particular, although I know the Arizona and Idaho are the two big ones, and then Berkeley runs some really good facilities, as does Lawrence Livermore. So there are facilities around the country that do great stuff. I am not familiar, though, at all with that one.
All right, and then I think our next question is, what about the story from about 3 weeks ago from Wisconsin rocks being found in Wyoming, supposedly transported by dinosaurs as what I would call gizzard rocks?
Yes. Okay. I'll just say it politely. I'm skeptical. In part, the gizzard rocks that people talk about, these gastroliths, are hard, and it's just they're very difficult to identify as truly being from an animal. The only time that I tend to go with the route that the only way I will believe something is with the gizzard stone, or is some sort of a gastrolith, right? A rock that's used to help break down food, just like modern birds and crocs do today. Is if you find it clumped together inside the skeleton. The problem is that those kinds of big clasts can be moved relatively easily. Now, the fact that you have some rocks that could be assigned to rocks from Wisconsin, they would be difficult to get to Wyoming. So it isn't disparage the research, because I have nothing to contribute other than I am skeptical of gastroliths as a whole, unless they are found in a skeleton. Beyond that, it does not mean that it's not a really cool question as to why are there rocks from Wisconsin, because the glaciers didn't go that way. So it's easy to get rocks from northern Canada into Wyoming, and that you can truly fingerprint to Wisconsin it's a viable hypothesis. I'll give them that. So,
And how long of a walk would that be?
oh,
At that time?
That time about the same. I mean, there was not that much difference in the continent. It didn't change that dramatically.
So
Depending
800,
on what time period they're looking at, because there was a minor seaway in between us at points in time. So they would have had to travel south and back up if we were in the Jurassic. But I don't recall where those rocks were found. So I'm not.
okay. But we're looking at 800, 900 miles.
Oh, yeah. Yeah, for sure. A long way.
And what do we know about the migration of dinosaurs?
Certainly that they migrated. Now there's I don't know. One of the things I love about paleontology, of course, you get to hear all my biases, is it's all the little stories that get told from one bone. And one of those stories is basically coming and look at isotopes, like strontium is an example. And they'll look at strontium isotopes, and basically organisms tend to like the lighter elements or the light isotopes and leave behind the heavy. And so you get these fractionations. Waters differ from place to place. And so you can look at a bone or a tooth that is growing for a period of time, and you can measure the amount of isotope, a particular series throughout that tooth or that bone, and you can see that strontium changes in organisms that migrate, for instance. And those that stay in the same local area and they're consuming the same water and food sources, the strontium stays very consistent. And so by using some of these isotope markers, we can trace at least the fact that animals migrated or they moved around a little. Now, to say how far they went, that's almost nigh on impossible to say unless we get something like gastroliths that we can truly fingerprint to one area and we're certain that that's where they came from. There's a lot of strongly supported. I mean, what's the saying? Extraordinary claims require extraordinary evidence. And
So Dave, your audio is breaking up more frequently now. So I think we ought to endure the barrel sound, if that's all right. Now it looks like you're frozen.
Yeah, it was on mine too.
Okay. And B, is this audio breaking up pretty frequently?
A little bit. I don't know if I noticed it quite as much as you did, but I did notice
Okay.
there was some
Yeah,
cuts in
we're
it.
gonna
I
have
didn't know
trouble.
I didn't know if it was my Wi-Fi or not. So I think that was my biggest worry.
Yeah. Well, I'm using I'm plugged in on the Ethernet, and it was breaking up quite a bit. Dareth, how was it, the audio? It looks like we lost.
Yeah.
Yeah, what I saw was that his internet connection was breaking up. Because
Okay.
I have a high speed internet here, 100 megabits per second that never has a problem. So
Okay.
it's on his end that he was losing internet connection.
Yeah, but I couldn't tell if it was between this headset microphone or if it's a Bluetooth. I didn't see a wire, so I
I
don't know.
couldn't tell what the source of the breakup was, but occasionally he would freeze in the image as well as have the sound break up.
Yeah.
Okay. Well, it's too bad because this is
Fascinating talk.
I think he's
yes,
back.
and okay, good. We'll have to troubleshoot this. So
Let's see, yeah. Maybe we can send them out if somebody's interested in that. Well, it doesn't seem like either Tom or Dave are having good luck logging back on, so I can't say if they'll be back on in time, but yeah, I guess that's all the updates I can give. I hope to see you all next week, and if anything changes, we can sure send out an email or maybe check back on the YouTube. If they do come back on and he answers some of them, I can put it into the end of the YouTube video of this if you have any questions that are unanswered.
Okay.
Thank you, Pia.
Thank you, Pia. Okay, I'll see you guys next week.
Yeah.
Take
Bye.
care. Bye-bye.